Hi class, and welcome to chapter 3. This chapter will all be on cells, and it's divided into 4 parts. And you'll see those 4 individual PowerPoints uploaded to Canvas. This lecture recording will get through parts A and B. Some of the parts will be a little review, so I might skip over those a little more quickly. The second part, part 3 or part C of this, is all on the inner organelles of the cell. And I'll have you guys review part C on your own. And then we'll spend a little more D, which is on description and translation. But this recording will go through parts A and B of chapter 3. And again, all of chapter 3 is on cells. So we need to really understand cells because the structure of the body's cells will explain why the permeability of the plasma membrane can affect the treatment. So what kind of treatment we give a person, whether that's drugs or some sort of antibiotic, a lot of it will have to do with these cells' membrane and understanding the permeability. So a little bit of cell theory. A cell is the structural and functional unit of life. How well the entire organism functions depends on the individual and combined activities of all its cells. The structure and function are complementary. That just means the structure of the cell will directly affect its biochemical function, and the biochemical function will be dictated by the shape of the cell and specific subcellular structures. Continuity of life has cellular basis, meaning that cells can only arise from other pre-existing cells. So there's over 250 different types of human cells, different size, shape, and components will give to differences in their functions. So this is a look at some of the different types of cells in the body. You can see erythrocytes are the red blood cells, epithelial cells are your covering cells, fibroblasts, skeletal muscle, smooth muscle cells, cells that store nutrients are your fat cells, cells that fight disease are your lymphocytes or macrophages, cells that gather information and control body functions are nerve cells, and then you have the cells of reproduction. So we have really diverse cells. So those different cells have some basic common features. A plasma membrane is the flexible outer boundary. The cytoplasm is intracellular fluid containing organelles. And then the nucleus is the DNA containing chemical center. And a lot of these basic parts of the cells, we were not going to touch on a lot. So I want you guys to review those on your own in this chapter. You can use the MyLab and Mastering, the website materials to review a lot of these organelles, more in detail about some of these organelles when we talk about transcription and translation and creating proteins. We'll talk about mitochondria and why they are important. But the basics of a cell, the plasma membrane, and all the organelles and what their functions are, you guys should review on your own and be sure you know them. Substances found outside the cells are what we refer to as extracellular materials. Classes of extracellular materials include extracellular fluids, like interstitial fluid. This is what cells are submerged or bathed in. Body plasma is the fluid of the blood, and the cerebrospinal fluid is fluid surrounding the nervous system organs. Cellular secretions are salivus and mucus that cells will secrete, and the extracellular matrix is the substance that acts as glue to hold the cells together. Is the active intracellular fluid from the extracellular fluid. So what is inside the cell versus what is outside the cell. It plays a very dynamic in cellular activity by controlling what enters and what leaves the cell. And it's also known as the cell membrane. The cell membrane consists of membrane lipids that form a flexible lipid bilayer. You have membrane proteins that float throughout this fluid, resulting in what we call a fluid mosaic model because it's constantly changing patterns. It's made up of many pieces. You have surface sugars that form the glycocalyx of the membrane, and membrane structures help to hold cells together through cell junctions. So this is a look at the lipid bilayer plasma membrane. You have different carbohydrates, proteins, lipids. Take a minute to pause, read through this to kind of review the lipid bilayer membrane of the cell. And here's just kind of a more zoomed in look at the lipid bilayer. Again, it's because it's made of 2 layers of these phospholipids. Again, the tails point to each other because they're hydrophobic, meaning they don't like to touch water. And the heads, the little blue circles, are hydrophilic. They love water, so they'll touch the exterior and interior environments of the cell. A great animation of membrane structure. Download this PowerPoint to look at these animations. This takes us through what the lipid bilayer is made up of, those hydrophilic and hydrophobic tails, some glycolipids, which are sugar groups, and then cholesterol, which increase the membrane stability. Here's a review of what phospholipids look like. And again, here is kind of a look at the intracellular fluid inside the cell, and the extracellular fluid is outside the cell. And you can see here, again, how the polar heads face the water inside and outside the cell, with the nonpolar tails hide from the water. Membrane proteins allow for cell communication with the environment. There's 2 types, integral proteins and peripheral proteins. Some float freely and some are tethered to intracellular structures. Integral proteins are firmly inserted into the membrane. Most are transmembrane proteins, which span the membrane, have both hydrophobic and hydrophilic regions, and they function as transport proteins, meaning as a channel or a carrier, also as a membrane or a receptor. Peripheral proteins are loosely attached. They function as enzymes, motor proteins for shape change, and also cell-to-cell communications. And this just looks at the many tasks of these membrane proteins, which again, I encourage you to pause the video and kind of review on your own. And this takes you through each of the tasks of some of these membrane proteins. They have tasks of transport, and this takes you through an animation of transport proteins. Receptors for signal transduction. This just means that a membrane protein exposed to the outside of the cell may have a binding site that fits the shape of a specific chemical messenger, like a hormone. And when that chemical messenger or hormone is bound, the chemical messenger may cause a change in shape in the protein that initiates a chain of chemical reactions in the cell. Another great animation on receptor proteins. Enzymatic activity. The membrane protein may be an enzyme with an active site exposed. A team of several enzymes in a membrane may catalyze steps of the metabolic pathway as indicated here. So something might bind to this enzyme and produce or be a part of a metabolic pathway. Cell-to-cell recognition. This just shows that glycoproteins, which are again proteins bonded to short chains of sugars, will serve as an identification tag that are specifically recognized by other cells. So those are some enzyme animations. Attachment to the cytoskeleton and the extracellular matrix. So elements of the cytoskeleton, which are the cell's internal framework or bones, and the extracellular matrix may anchor to membrane proteins. So providing some sort of attachment to what's on the exterior of the cell, to what's on the interior of the cell. This helps to maintain cell shape, fixes the location of certain membrane proteins, and plays a role in cell movement. Cell-to-cell joining, so membrane proteins can help cells to join together. These membrane proteins can be called cell adhesion molecules, or CAMs for short. Glycolytics then consists of sugars sticking out of the cell surface, and every cell type has different patterns of this sugar coating, and this functions as specific biological markers for cell-to-cell recognition, and it allows the immune system to recognize the self versus non-self. So this will allow your body cells to recognize something as non-self. Clinical homeostatic imbalance, the glycocalyx of some cancer cells can change so rapidly that your immune system cannot recognize the cell as being damaged. So the mutated cell will not be destroyed by the immune system, because the body can't recognize it as being damaged. So it's able to replicate extremely quickly before the body can damage it or destroy it. Cell junctions, so some cells are free, meaning not bound to any other cells. Examples, blood cells and sperm cells are free. Most cells are bound together to form tissues and organs, and there's 3 ways in which these cells can be bound together: tight junctions, desmosomes, and gap junctions. And I'll let you guys review these junctions in the body. The tight junctions are shown here. They're an impermeable junction. They form a continuous seal, so they will prevent any molecule from passing between the cells. Desmosomes are rivet-like junctions that are formed when linker proteins of neighboring cells interlock like teeth or a zipper. These kind of form plaques or button-like plaques. So this is a look at desmosomes. They're anchoring junctions. They bind adjacent cells together like Velcro, so they help keep cells from tearing apart. And then gap junctions have transmembrane proteins that form tunnels that allow small molecules to pass from cell to cell. And this is used to spread ions, sugars, or other small molecules between the cells. So this will allow for an electrical signal to be passed quickly from one cell to the next. And this is used in cardiac and smooth muscle cells. So here's a look at gap junctions. It allows communication. So it allows ions to pass through, particularly important in heart cells, embryonic cells, with the passage flow of ions. The cell junctions, the plasma membrane is selectively permeable, allowing only certain molecules to cross it. And there's 2 essential ways substances can cross the plasma membrane: either by passive transport, meaning no energy is required for things to pass through the plasma membrane, or by active transport, where energy, ATP, is required. Passive transport requires no energy input. There's 3 types of passive transport: simple diffusion, facilitated diffusion, and osmosis. And you should have a good understanding of these 3. All types involve diffusion, which is the natural movement of molecules from an area of high concentration to an area of low concentration. This moving down of a concentration is referred to as a concentration gradient. And a good example of this is putting creamer in your coffee, where the creamer first starts, it's very concentrated, and then it will naturally diffuse to an area of high concentrated coffee creamer to low concentration throughout your entire cup of coffee. So that's what diffusion is, moving from an area of high concentration to an area of low concentration. So this can happen in liquids, it can happen in air. I usually give the example, if I would spray some perfume in the front of the classroom, eventually those perfume molecules would diffuse throughout the entire classroom, so everyone would be able to smell it. So all molecules have this random high-speed movement due to their intrinsic kinetic energy. And that just means that the movement between particles, it results from collisions between molecules. So molecules in higher concentrated areas will collide more, resulting in molecules being scattered to lower concentrated areas. So this is just a look at diffusion. The concentration gradient is the difference in concentration from an area of high concentration, for example, of this dye pellet, to low concentration of the dye. And as time moves on, the random thermal motion, the kinetic energy, makes the dye molecules move and collide constantly. And as a result, they move their concentration gradient down from an area to high to low concentration, until eventually the dye is evenly distributed, where there's no more net movement of the dye because there's no more concentration gradient. The speed of diffusion is influenced by the concentration of the 2 areas. So the greater the difference of concentration, meaning if you put a more strongly concentrated dye, the faster diffusion occurs. Molecular size, smaller molecules diffuse much faster than larger ones, and temperature. Higher temperature increases the kinetic energy, so it increases the molecules bouncing off of each other, which results in faster diffusion. Equilibrium is only really reached when there's no net movement of molecules, and the net movement is in both directions. So diffusion, molecules have a natural drive to diffuse down their concentration gradient. And in terms of the cell, the nonpolar hydrophobic lipid core of the plasma membranes will stop diffusion and create concentration gradients by acting as a selectively permeable barrier, meaning it only allows certain things to come through. Molecules that are able to passively pass through the membrane include lipid-soluble and non-polar substances, very small molecules that can pass through membrane or membrane channels, and this is referred to as simple diffusion. If a larger or non-lipid soluble or polar molecule needs to cross over, and it referred to as diffusion. Facilitated diffusions don't require any energy by the cell, but it usually requires the assistance of some sort of carrier molecule to get the substance through. Osmosis is a special name for the movement of solvent, usually water, not molecules. So osmosis just means that water is moving through diffusion. If the plasma membrane is severely damaged, substances will diffuse freely into and out of the cell, compromising the concentration gradient. So for example, a burn patient, if their cells are significantly damaged in their skin, their cells will lose precious fluids, proteins, and ions that will weep. That just means they will exit from the damaged cells. So simple diffusion then, nonpolar lipid-soluble hydrophobic substances can diffuse directly through the phospholipid bilayer of the cell membrane. So examples of this, oxygen, carbon dioxide, steroid hormones, and fatty acids can freely pass through the cell membrane. And this is a great animation of diffusion that please watch on your own. This is diffusion through the plasma membrane, simple diffusion. So solutes, lipid-soluble solutes. So again, they're lipid-soluble, meaning they have to pass through the lipid membrane, and they're moving from an area of high concentration to low concentration. Facilitated diffusion then, certain hydrophobic molecules like glucose, amino acids, and ions are transported passively down their concentration gradient, but they need a little help to get through the membrane. So we have protein carriers that these substances can bind to get through the membrane, or there's channels that substances can move through. Carrier-mediated facilitated diffusion just means that carriers, they are transmembrane integral proteins, and they will transport specific polar molecules like sugars and amino acids that are too large for the membrane channels. Binding is limited by the number of carriers present. So if a carrier is saturated, that just means it's all bound to molecules that are busy transporting. So this is just an example of a carrier, which will help some sort of lipid insoluble solute. So it's not able to pass through the membrane. So it'll go through by way of a carrier. And again, this is facilitated diffusion. It still does not require any energy by the cell. It'll happen passively. So travel from an area of high concentration to low concentration. The channel-mediated facilitated diffusion, the channels are aqueous-filled cores, and they're formed by transmembrane proteins. The channels will transport molecules such as ions or water through osmosis down the concentration gradient. Water channels are called aquaporins, and these channels are based on pore size and charge of ions present. We can have leakage channels, which are always open, or gated channels, which are controlled by chemical or electrical signals. So this is a look at diffusion through the plasma membrane through channel-mediated facilitated diffusion. Osmosis then is just the movement of solvent, not molecules. So the osmosis is the movement of water across the selectively permeable membrane. Water will diffuse across the plasma membrane through the lipid bilayer. Even though water is polar, it's so small that some of the molecules can still sneak past the non-polar phospholipid tails, or through specific water channels called aquaporins. The flow will occur when water concentration is different on two sides of the membrane. And I'll explain this for a little bit. So water can travel through aquaporin channels, or if it's really tiny, it can travel through the lipid bilayer kind of one at a time. And this is osmosis, diffusion through the plasma membrane, the movement of water. Again, it's still passive. It'll happen without energy down its concentration gradient. Osmolarity is the measurement of the concentration of the total number of solute particles in a solvent, and the water concentration will vary with the number of solute particles because solute particles will displace water. So what this means is when solute concentration goes up, the water concentration will go down. So water will always move by osmosis from an area of low solute or high water concentration to an area of high solute or low water concentration. So what this just means is it just means that water will always move into an area that's more highly concentrated with solute. And this makes sense if you think about kind of a salt water, or if you put salt or sugar into a solution, water will always want to move into an area where there's higher solute solution to try to disperse some of that solute, that salt or sugar. When the solutions of different osmolarities are separated by a membrane permeable to all molecules, diffusions of solutes and osmosis of water will occur, and they will cross the membrane until equilibrium of solutes and water is reached. And equilibrium just means that the same concentration of solutes and water molecules on both sides with equal to volume on both sides. So an example of this, if we have a membrane permeable to both solutes and water, that means that both water can get through and both solute particles can get through. For example, sugar. What will happen is solutes and water will pass through this freely permeable membrane until equilibrium is achieved, meaning both sides have the same osmolarity or concentration. The volume will be unchanged because both water and solutes can travel freely through this membrane. So both solute and water molecules will move down their concentration gradient. So if you look here, the sugar molecules are more concentrated on the right side of the membrane. So sugar will move down its concentration gradient to the left until there's equal concentration. And water is going to move in an area of the sugar molecules because it will travel down its concentration gradient. Osmosis, once a little more on osmosis, when solutions of different osmolarities are separated by a membrane that is permeable only to water, only osmosis will occur until equilibrium is reached. So water will have a net movement across the membrane until osmolarity is the same on both sides, and this will result in volume changes on the both sides. And I'll show you an example of this, but the low solute side volume will decrease and the high solute volume will increase. So what do I mean by this? So now we have a membrane which is permeable to water, but impermeable to solute. So this means that this membrane, only water can pass through. So we have this U-shaped beaker holding water and sugar molecules. More sugar is concentrated on the right side than the left. Sugar at this point is unable to travel through the membrane. So only water is able to travel through, and water will always travel into an area where there's more solute particles. And when water does that, when water moves to an area of higher solute concentration over to the right, we see this volume change. So we get to equilibrium because at equilibrium, we have the same osmolarity because water moved into an area of higher concentration, but we have different volumes of water on both sides of the beaker. And that's because we need more water on the right side to spread out the more solute molecules to get the equilibriums of the concentration to be equal. So again, water moved into an area of higher solute concentration. So that is osmosis, water moving into an area of greater solute molecule concentration to eventually get to equilibrium. And you can see here, because we have more volume now on the right side, there is an equilibrium of concentration or osmolarities between both sides of the membrane. Hydrostatic pressure is the outward pressure exerted on the cell side of the membrane caused by an increase in the volume of the cell due to osmosis. So the movement of water involves these two different types of pressures, and the osmotic pressure refers to the inward pressure due to the tendency of water to be pulled into a cell with higher All right, so these are the two pressures that water involves through osmosis, and you can read through some of these pressures on your own. This hydrostatic pressure and osmotic pressure is just important. especially for plant cells, so that it kind of gives them their strong cell wall, so that the plant cell will fill with only so much water and then stop. Animal cells don't have this cell wall, so they cannot limit the amount of these pressures on the cell wall. So animal cells can burst if too much water is taken in. Water can also leave the cell, causing it to shrink. So changes in the cell volumes can disrupt cell functions greatly. And this brings us to what we call tonicity, which is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. An isotonic solution has the same osmolarity as the inside of the cell, so volume remains unchanged. A hypertonic solution has a higher osmolarity or concentration than the inside of the cell. So water will flow out of the cell into an area of higher concentration into that hypertonic solution, causing the cell to shrink. We call this shrinking of the cell crenation. A hypotonic solution has a lower osmolarity than inside the cell. So the water will flow into the cell, resulting in the cell swelling, and this can lead to the cell to burst, referred to as lysing. This will take us through a little bit of osmolarity. It's equal to the molarity times the number of ions. And we're kind of going to skip over this. You guys don't have to worry about too much of this, but I do want to talk about the effect of solutions of varying tonicities on living red blood cells. And you should have a good understanding of this. So if we were to place a red blood cell in an isotonic solution, that just means that the osmolarity of the inside of the cell would be the same as the outside of the cell. So there'd be no net movement of water. Water will freely move into and out of the cell, but the cell will maintain its shape because it's in a solution of the same tonicity or same concentration. However, if we put a red blood cell in a hypertonic solution, so in a solution that has a greater concentration surrounding it, because of osmosis, water will naturally travel into an area of higher concentration. So water will actually travel out of the cell into that hypertonic solution. And this causes the cell, the red blood cell, to shrink. And we call that crenation. And this is what happens when you drink salt water, or if you were to drink salt water, you bathe your cells in that really hypertonic, really highly concentrated solution. So water will leave your cells, causing them to shrink, and you'll become dehydrated. In a hypotonic solution, so if we place the red blood cell in a hypotonic solution, so in a solution that's less highly concentrated, what will happen is water again travels into an area of more concentration, which in this case would be inside the cell. And this can cause the cell to swell and possibly burst. And we call that cytolysis if the cell actually bursts. Here's a great animation on osmosis. And we are I'm going to take us through kind of the next PowerPoint. If you're still with me, I'm going to share part B, and we'll get through part B a little bit quicker here, and then we'll be done with this lecture recording. So let's go ahead and start on part B. So again, we're still talking about cells, a little bit more about transport processes. We talked about the passive transport processes at the end of part A, and now we're going to talk about just active transport processes. So the two major active membrane transport processes are active transport and vesicular transport, and both require ATP or energy to move solutes across the plasma membrane for any of these reasons. If the solute is too large for a channel, the solute is not lipid soluble, so it can't make it through the membrane on its own, or the solute is not able to move down its concentration gradient. So for example, if we want to get a solute particle or a substance to move against its concentration gradient, so move back into an area of higher concentration, it requires active transport. So active transport requires carrier proteins or pumps. Some carriers transport more than one substance at a time, and an antiporter transports one substance into the cell while transporting a different out. A symporter transports two different substances in the same direction, and both will move solutes against their concentration gradient, so an area from low to high concentration. Think of trying to trying to get into a bus that's just jammed full of people. You're moving against your concentration gradient because you're going from an area of less concentrated people into an area of more. So that will require energy. Primary active transport requires energy coming directly from ATP hydrolysis. That means the breaking up of those phosphate bonds. And secondary active transport, the required energy is obtained indirectly from ionic gradients created by primarily active transport. So primary active transport is energy from the hydrolysis of those ATP causing changes in shape. An example of primary active transport are different pumps, especially the sodium potassium pump that we're going to learn a lot about in this class. The sodium potassium pump is the most studied. It's basically an enzyme called sodium potassium ATPase that pumps sodium out of the cell and potassium into the cell. It's located in plasma membranes, but especially it's located in excitable cells like nerves and muscle. Leakage channels located in membranes result in leaking of sodium into the cell and leaking of potassium out of the cell. And the sodium potassium pump works as an antiporter that pumps sodium out and potassium in against their concentration gradients. And this is important for maintaining what we call an electrochemical gradient, which involves both the concentration and electrical charge of ions. And this is extremely essential for functions of muscle and nervous tissue. So here's a look at primary active transport, the sodium potassium pump. You can kind of read through this on your own, but basically what it's doing, it's pumping two you can see the 3 potassium coming into the cell, or excuse me, this 2 potassium coming into the cell and the 3 sodium going out of the cell. And we'll learn a lot more about this when we get to the nervous system. Step 1, 3 cytoplasmic sodium bind to the pump protein. So they're coming from inside of the cell. Sodium binding promotes hydrolysis of ATP. The energy released during this reaction phosphorylates the pump. Phosphorylation causes the pump to change shape, which expels the sodium to the outside. So we're pumping 3 sodium to the outside of the cell against its concentration gradient. 2 potassium on the outside of the cell bind to the pump. Potassium binding triggers release of the phosphate, and the dephosphorylated pump resumes its original conformation. The pump protein binds ATP and releases potassium to the inside, and the cycle repeats again. So this takes us is a good kind of basic introduction into what we call resting membrane potential. So watch that video and we'll talk a little bit about it. Secondary active transport depends on the ion gradient that was created by primary active transport. The energy stored in gradients is used indirectly to drive transport of other molecules. So the low sodium concentration that is maintained inside the cell by the sodium potassium pump will strengthen sodium's drive to want to enter the cell. Sodium can drag other molecules with it, also called a symporter. So some sugars, amino acids, and ions can be usually transported into cells via secondary active transport. And this just shows the secondary active transport is driven by the concentration gradient created by the primary active transport. And you can see that here. So glucose will come in with sodium because of the gradient that is being established. Vesicular transport involves transport of very large molecules. So this is the second part of active transport, how we get things into and out of the cells. It also requires energy. And this is when very large particles, macromolecules, and fluids have to be moved across the membrane in membranous sacs called vesicles. Endocytosis is transport into the cell. And there's 3 types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And exocytosis is the transport out of the cell. Transcytosis is transport into, across, and then out of the cell. And vesicular trafficking is the transport from one area or organelle in the cell to another. Endocytosis involves the formation of protein-coated vesicles. It involves receptors, so it can be very selective. The substance will be pulled in and must bind to its unique receptor. Some pathogens are capable of hijacking this receptor for transport into the cell. And once the vesicle is fused into the cell, it can be fused with a lysosome or undergo transcytosis. So it can undergo some sort of transformation or fuse with a lysosome, which helps with the degradation or getting rid of it. So this is a look at endocytosis. So a coated pit will ingest a substance. The protein-coated vesicle will attach. It will undergo some sort of transportation. Coated proteins will be recycled to the membrane. It will either be taken up by a lysosome, so stored. The fused vesicle can fuse with the lysosome for digestion. The transport vesicle containing the membrane components will move back to the plasma membrane for recycling. And then it will possibly deliver its contents to the plasma membrane of the opposite side for transcytosis, so moving across the cell completely. And this takes you through the different steps that you can kind of watch on your own. And again, feel free to pause this and watch it on your own. Phagocytosis is a type of endocytosis that is referred to as cell eating. So the membrane projections called pseudopods form around the solid particles, and it engulfs, forming a vesicle being pulled into the cell. So this is phagocytosis, is cell eating, so engulfing a larger particle, usually a solid particle. Pinocytosis is cell drinking, where it usually enfolds a smaller particle or a fluid or a liquid. So that's pinocytosis, something smaller. Receptor-mediated endocytosis involves an endocytosis and transcytosis of specific molecules. Many cells have receptors embedded in what we call clathrin-coated pits, which will be internalized along with the specific molecule bound. So this is a look at receptor-mediated endocytosis, so binding to a receptor first. And then exocytosis is the process where material is ejected from the cell. It usually is activated by cell surface signals or changes in membrane voltage, where the substance being ejected is enclosed in a secretory vesicle. The protein on the vesicle is sometimes called the V snare. It'll find and hook up to a T snare target protein. And this is just the docking process that will trigger exocytosis. And things that will be kind of exocytosed from the cell will be hormones, neurotransmitters, mucus, or cellular wastes. So this is a look at exocytosis. And this picture, so this is a look at kind of the V snare will dock to the T snare to show kind of the process of fusion. And then it will be exocytosed when a fusion pore is formed. And then the vesicle contents will be released to the exterior side. This is a really cool photomicrograph of about 100,000 times magnified of a secretory vesicle releasing its contents via exocytosis. It's always fun to see like a real life micrograph of this, to see that this is actually something that happens in real life. So this brings us to resting membrane potential. This is the electrical potential energy produced by separation of oppositely charged particles across the plasma membrane in all cells. The difference in electrical charge between 2 points is referred to as voltage, and cells that have a charge are said to be polarized. Voltage occurs only at the membrane surface. The rest of the cell and the extracellular fluid are neutral, and the membrane voltages range from about -50 to -100 in millivolts in different cells. And the negative sign just indicates that the inside of the cell is more negative relative to the outside of the cell. The ion potassium is a key player in resting membrane potential. It will diffuse out of the cell through potassium leakage channels down its concentration gradient. Negatively charged proteins cannot leave, so as a result, the cytoplasmic side, so the inside of the cell membrane, becomes more negative. Potassium is then pulled back by the more negative interior because of its electrical gradient. And when the drive for potassium to leave the cell is balanced by its drive to stay, the resting membrane potential is established. And most cells have a resting membrane potential around -90 millivolts. That's kind of a number you should remember. The electrochemical gradient of the potassium then sets the resting membrane potential. In many cells, sodium also reflects the resting membrane potential because sodium will be attracted to the inside of the cell because sodium is positive and it will want to come into the negative charge. So if sodium enters the cell, it can bring the resting membrane potential up to around negative 70 millivolts. Then the membrane is usually more permeable to potassium than sodium. So potassium will actually be the primary influencer on the resting membrane potential. So this just shows how potassium will diffuse down its steep concentration gradient. So potassium will move into the cell because it will be attracted to the negatively charged established of the inner membrane potential. And a negative membrane potential is established of about negative 90 when the movement of sodium out of the cell equals the movement of potassium into the cell. And at this point, the concentration gradient promoting potassium exit will exactly oppose the electrical gradient. So what I just want you to know here is that we have ion channels that create a resting membrane potential of about negative 90 millivolts inside the cell. So the cell is always usually a little more negative right inside the cell at the membrane. The resting membrane potential is also maintained through the sodium-potassium pump, which will continuously eject 3 sodium out and bring 2 potassium in. So this will kind of keep the resting membrane potential where it should be. A little bit about cell-environment interactions. Cells will interact with their environment by responding directly to other cells or indirectly to extracellular chemicals. Interactions always involve the glycocalyx, which includes cell adhesion molecules or CAMs and plasma membrane receptors. Every cell has thousands of sticky glycoprotein CAMs projecting themselves, and these are their functions: anchor the cells to each other, assist in movement, attract white blood cells, stimulate the synthesis or degradation of adhesion proteins, transmit intracellular signals. Membrane receptor proteins serve as binding sites for several chemical signals, contact signaling, chemical signaling. These are just different roles of membrane receptors. Chemical signaling is important because the chemical ligands can bind, causing an interaction inside the cell. This can activate what we call G protein linked receptors, and they can in turn activate G proteins, which can in turn affect ion channels or activate other enzymes or cause release of internal secondary messenger chemicals such as cyclic AMP or calcium. So this is just the role of plasma membrane receptors. When they will bind to something, it could cause some sort of cascade reaction inside the cell that will focus on when we get to some of the physiological processes of the body. G proteins are important. They act as a middleman or a relay between extracellular first messengers and intracellular second messengers that cause responses within the cell. And this takes you through how the G proteins will interact. So the ligand, the first messenger, will bind to a receptor. The activated receptor will bind to a G protein and activate. The activated G protein will activate an effector protein by causing its shape to change. And then the activator enzyme will catalyze a reaction that produces a second messenger inside the cell. And the second messenger will then go on and activate other enzymes or ion channels. The kinase enzyme will activate other enzymes, and that takes you through a brief interaction of those G proteins, which we will see later throughout the semester. Again, this was the first half of chapter 3, and I will post the second half recording a little bit later. Thank you for listening, guys.